For chapter 17.2, we're going to talk about mapping genomes. So we're going to need to talk about genomics to talk about this. Then we'll need to discuss physical versus genetic maps. And then we'll need to talk about how maps are actually made. We've already kind of looked at this in the genetics chapter, but this is an important stepping stone on our way to understanding how the human genome was sequenced. So remember, genetic maps are an order of genes on chromosomes. So genetic mapping is just finding that order of genes on chromosomes. And that was done initially using recombination and linkage. So understanding that two genes were linked showed us that they were closer than another gene. So this could be done by either looking at the physical traits themselves or using what we call genetic markers. A genetic marker is a specific sequence of DNA that is linked to a gene of interest, so like eye color. And you know that this piece of DNA tends to move with eye color, and this specific SNP or something like that moves with eye color. So the earliest genetic maps were done in fruit flies, and we saw that different traits like eye color, here we have purple versus red eyes, or wing shape, or antennae little size, or things like that. Different traits were linked together to create a map. And this is great, but we care a lot about humans because we are humans. So looking at a human genetic map, we find it to be rather lacking, right? We estimated there were anywhere between, 30,000 and 100,000 human genes before we sequenced the human genome. But here's a human gene map of a specific chromosome. So look at these different genes that are on here. What's kind of the common theme between them? Go ahead and look at it. Think about that. All right, well, many of these look like Manx syndrome, Alport syndrome, Duchenne muscular dystrophy, hemophilia A, Fragile X syndrome. None of these things sound very good, do they? They sound like genetic diseases. So our initial human genetic maps were often very, very focused on genetic diseases because those were easy traits that we could map. Okay, so we did have genetic maps, but how do we figure out what chromosome they're on and where on that chromosome they actually lay? Well, that's our next step in the process, and that involves creating what we call physical maps. A physical map deals with the actual location of the gene on the chromosome, and to do that is a little bit tricky and involves a process that we call hybridization. So we're going to look at this process now. So fluorescent in situ hybridization, or FISH, is a common method for identifying where a specific sequence of gene is on a chromosome, and it relies on complementary base pairing. So we take what's called a DNA probe and we label it with a fluorescent dye. We then allow the probe to hybridize or match up with the DNA sequence. So we make this probe specifically to match a sequence of DNA we care about. Then we apply certain conditions to the chromosome and allow this fluorescent probe to match up with it, showing us an actual location on a chromosome. So FISH was really important in saying, okay, we know these genes are linked, but what chromosome are they on? FISH could help tell us that. Okay, these fluorescently labeled DNA sequences that match a gene, what did we call those? Pause the video, think about that. Okay, we called these probes. We're probing to find whether they can match up together. So this allowed us to build our first maps here, physical maps. So genetic mapping using recombination to determine the order of genes was being done, but we supplemented that with genetic markers to help tell us sequences of DNA that were close. So once we could start like genotyping, we could start figuring out these things at a molecular level. But human genetic maps tend to be incomplete, and physical maps are necessary to show us which chromosome the genes are actually located on. To do that, we use fluorescent in situ hybridization, which uses labeled DNA probes to attach to the chromosome locations. All right, in the next section, we're going to take this information and move on to sequencing the whole genome.